Okay, maybe let's start off right now with a poll just to get everyone kind of going and thinking about the last lecture and some of the material we went over in lecture 9. So I'm going to just give you a very basic poll just so we can get, you know, kind of interactive a little bit. So the tata box binding protein helps RNA polymerase to recognize promoter. Which of the following is correct or based on the way that this TBP normally binds to the promoters?
What does the last option say? Because on my side, it's kind of like cut off.
Binding of TBP to promoters is not influenced by nucleosome positioning.
Okay, thank you.
Okay. Let me go ahead and end this. Okay, could you guys see the results?
Yes.
Okay, so as you can see, again, it's all over. The majority of you did say that TBP binds to the minor groove of the tata box, and that is correct, right? So if you think about the TBP, there was a slide specifically showing that, how it really hooks into that minor groove of the DNA and forms like an anchor. And that's kind of very special to TBP and the way it interacts with DNA at the promoters. So A is not the option, right? It doesn't bind to the major groove, it binds to the minor groove. And some of you also noted that it binds only to promoters with a tata box. That's also not true, right? It can be recruited there with the tata box associated factors, the TAFs, right? The TAFs recruits the whole complex to promoters that don't have a tata box, right? So it either have an initiator element or a DPE element or a GC box, right? So TBP can still get to promoters in that way, okay? And binding to TBP to promoter is not influenced by nucleosome positioning. And that is kind of, we haven't discussed this so far. Yeah. Any questions about that? Okay, so let's start today, just a refresher on some of the ideas behind chromatin structure, right? So lecture 8 and lecture 9, we really focus on transcriptional gene regulation and how the eukaryotic gene expression is regulated, right, either by the polymerase, RNA polymerase 2, and all of the factors that's involved in associating at a promoter and recruiting RNA polymerase. And then we also talk about these activator elements, which are also important for regulating gene expression beyond that basal level of transcription that you normally get. Another really important aspect of gene regulation of gene expression in eukaryotes is chromatin structure. And that's primarily because the DNA within eukaryotes is not a naked molecule, right? It's often bound by multiple additional proteins. It's also compacted into a state where it can fit within the nucleus. And all of these proteins play a role in regulating the accessibility of that DNA to the transcription machinery, okay? And these proteins are used, you'll see in a minute, right? It's either histone proteins, which form this basic unit of the nucleosome along with the DNA. And then you also have a range of other proteins that are also important for regulating this chromatin structure. So we want to remember that the core nucleosome unit is this structure that includes a histone proteins, right? So 4 histone proteins make up this histone octamer. You also have the DNA that's wrapped around this histone core. And then you have a region of linker DNA that's also available, right? So if I ask you what's the core nucleosome unit, you want to make sure that you understand that this is composed of these histone core proteins, right? About 146 base pairs of DNA wrapped around, and also 20 to 60 base pairs of linker DNA. This is really the fundamental component of a core histone unit, okay? Within that unit, right, you have these histone core proteins that form dimers and tetramers among the 4 units, right? So you have an H2A and H2B dimer, and you have 2 of those, right? So they're both here. And then you also have an H3 and H4 tetramer, okay, that's present here. The DNA gets wrapped around those histone proteins. What's sticking out of this nucleosome unit are the tails of those histone proteins, the amino terminal tails or the N-terminal tails that are often sticking out of that nucleosome unit. And this is important because the modification of these histone tails is really what helps to facilitate changes in chromatin structure and ultimately regulate gene expression. The way that the DNA is wrapped around the nucleosome is also important. The histone core proteins are also important, right? So you have the DNA bent around, and the core proteins make contact with the minor groove of the DNA, and that's primarily AT-rich, right? And this, of course, is important because AT-rich has 2 double bonds compared to a more GC-rich sequence. So it helps to facilitate easy dissociation from the histone proteins when needed. So that's something that you also want to think about, how these proteins are making contact with the DNA in this structure. In addition to the core histone proteins and the way that the DNA gets wrapped around it, you have an important role for histone H1, which is a linker protein. This linker protein is important for helping to keep the DNA tightly bound around the histone proteins, but it also helps to organize the way that the DNA is wrapped around the histone core, right? So there's an order in how the DNA gets wrapped around. It's just not randomly wrapped around these histone proteins, right? There's a really defined angle of entry and also an exit, right? Because this needs to be regulated. So when you need to remove that DNA from those proteins, it also needs to happen in a particular order. So it's highly regulated. And that's where these histone H1 linker proteins play a role in that regulation. We also discuss ways in which, and this is just a refresher from the last lecture, we discuss ways in which those histone proteins can affect gene activity, right, on its own. So you have your 30 nanometer fiber, which is where you have the chromatin in a compacted state. And the orange ovals that you're looking at here is representing the linker proteins. So you can have a linker protein that's really much preventing access of this transcription start site or the promoter of that piece of DNA. And that's what this arrow is representing. When this access is blocked, gene expression is repressed, right? And this is the other option is you can also have an entire nucleosome unit over the promoter and over the transcription start site. So you're not able to get the transcription machinery to this site. So in both of these cases, whether it's the linker protein or an entire nucleosome that's blocking the transcription, the gene will be in the repressed state. What can happen is that you can have removal of that histone, that linker protein, facilitating gene expression, because now you have access to DNA, you can recruit an activator and you can have gene expression. You can also evict the nucleosome from this location, right? So you have the linker protein is removed, then you have additional proteins such as chromatin remodeling complex, which we will talk about next, will come in and remove this nucleosome from this transcription site, and then activators can get recruited and you can have gene expression, right? So that's kind of how the linker proteins help to affect gene activity. We also discussed that you need these chromatin remodeling complexes, which are huge protein complexes that comes in, recognizes the regions of this chromatin and helps to remodel the chromatin, right? Making it either more accessible for transcription or less accessible, right? Depending on the chromatin remodeling complex and all of the different proteins that they might interact with. What's important to know about these chromatin remodeling complexes, they're highly ATP dependent, right? They need ATP to function. And they also help to change the contact between the histones and the DNA, right? So they're really facilitating either more association or less association. There's different ways in how the chromatin remodeling complex can remodel these chromatin. And depending on the chromatin remodeling complex, you will be able to facilitate one of those types of methods of remodeling the chromatin. So the SWI/SNF group of chromatin remodeling complex are more often involved in either remodeling of the way that the DNA is wrapped around the histone proteins, or by completely displacing the proteins from the DNA, okay? So depending on the family of these SWI/SNF or the complex that's present, you can have one of these two possibilities happening. The other group that's also important, and we talked about three of them, right? So this is the second group that we also discussed, which is imitation switch chromatin remodeling complex. They help to slide the DNA away from the histone proteins, or actually in a different position of the histone proteins. So you can imagine what I mean by that. When you're sliding, if you're pulling on this end, right, you're pulling on this end, maybe you're making some of the DNA here that would have been blocked by this histone protein more accessible. But you can also imagine that you're also making this accessible DNA more blocked, right? And imitation switch chromatin remodeling complex is actually most often associated with gene repression, right? So it makes the DNA less accessible. But it can be both. The other chromatin remodeling complex we discussed was SWR1, which is a chromatin remodeling complex that plays a role in replacing the core histone with a histone variant. And by doing so, if in this case, if you're replacing H2A with H2AZ, which is a histone variant, the association with the histone variants to the DNA is not as strong. So it's much more easy for you to move the DNA away from those histones. So it makes it's a more loose architecture with the histone variants when DNA is wrapped around. Okay, so those are the three main chromatin remodeling complexes that we discussed. Understanding what each of those complexes do, understanding that you absolutely need ATP for this to work will be important. And similar to the ways that we have shown how you can determine whether or not you have a protein binding directly to the DNA, there's a very similar technique which includes using a nucleus to determine whether or not a particular piece of DNA is wrapped around the nucleosome. Okay, and this technique is called MNase-seq. MNase is the enzyme, is micrococcal nucleus from Staphylococcus that's being used in this case, and it's able to digest the DNA between two nucleosome units. Okay, so in this case, you have a nucleosome, you have the DNA wrapped around it. It's pretty much cutting within this linker region. Okay, and cleaves the DNA right on each side. Once you add this MNase treatment, it's a highly sensitive technique. You have to know how much MNase you're adding. So it's a highly sensitive technique, but once you're able to optimize it, you're really able to get individual nucleosomes isolated. So using this MNase treatment, what you're left with is the nucleosome and the DNA that's wrapped around it. Okay. So what this allows you to do is say, okay, can you determine now whether or not this DNA or any piece of DNA was wrapped around the nucleosome? At this step, the only thing that you will have in your solution will be the DNA that's wrapped around a nucleosome. Everything else has been chewed up, right? So then you can do some other alterations to the solution to break the connection between the nucleosome, the histone proteins, and the DNA, and then isolate the DNA. Once you isolate the DNA, again, this is genome-wide, you sequence it using a high throughput sequencing method, and you can determine in your genome which DNA, I seem to be having this problem again, which particular DNA was associated with a particular nucleosome, okay? So it's really a powerful technique to particularly look at nucleosome positioning, okay? Within a cell. I hope that this doesn't, I'll try this one more time. I had the PDF prepared because I was ready for this. We've all been having issues with a 2016 version of these Apple computers. So we're trying to figure out what to do with those. Just bear with me. I have the PDF, so if it crashes again, we'll just use the PDF. Yeah, so this technique, as mentioned, is very powerful for you to determine nucleosome positioning, okay? It's MNase-seq compared to DNA-seq, right? DNA-seq, which tells you whether or not a particular protein is bound to a particular DNA. Similar to footprinting assay, right? We talked about footprinting assay, which will also tell you whether or not a protein is bound to a DNA, and you're using an enzyme as well to chew up the DNA that's exposed. And so it's kind of a very similar technique when you think about it. So the way that the way that histones are considered to regulate gene expression is what we refer to as this histone code hypothesis. And this really is a hypothesis that's based on the fact that the modification of the histone tails will determine how a gene might be regulated, whether it's going to be expressed or not, okay? And as I mentioned, you have these histone tails that's sticking out from the different histones. What gets modified is the different amino acid residue within that histone tail. So these modifications are considered post-translational modifications, right? Because you're modifying the amino acids. The position of the amino acid also determines whether or not a gene will be activated or repressed, depending on which modification happens, okay? And it also depends on which histone protein gets modified, which of the histone tails get modified. Three of the major types of histone modifications where we know a whole lot about in terms of how they regulate gene expression is methylation, phosphorylation, and acetylation. As I mentioned, depending on which amino acid residue gets modified, it will determine whether or not a gene is expressed or not. One of the ones that's been very well studied in terms of the modification is histone protein 3, so H3. We know a lot, we understand a lot about the different modifications on the tail of histone 3. Depending on which residue gets modified, first sometimes, right? So here, if you can see, this is lysine, which is represented by a K. So lysine 9, so histone H3, lysine 9 can be methylated or acetylated, right? And depending on whether or not it's methylated or acetylated, it can determine which one it is, okay? So these modifications of these histone tails, the way that they're able, and you'll see in a minute, so to answer your question, Christian, right? It really depends on what proteins they're able to recruit, okay? So the tails get modified and they will recruit certain types of chromatin remodeling complex and different types of proteins that will then direct whether or not the gene will be activated or repressed. So knowing, you know, that these, depending on the modification, you can have either a positive or negative gene expression is important to know, right? So acetylation of pretty much most of the, I would say pretty much all of the lysines that we are aware of is associated with activation of gene expression. Okay, so histone H3, lysine 9, 14, or 27, any of these three is acetylated, you have activation. Methylation varies, right, depending on which lysine gets methylated and also how many methyl groups are added to that site, right? So you can have a monomethylation, dimethylation, or trimethylation. Depending on what you have, you can have different types of effect on gene expression. You will be required to know a activation mark and a repression mark, right? So you can decide however you want to think about this, but you will need to know one of these two marks that's involved in either positive regulation or negative regulation of gene expression. So the way that acetylation is able to affect gene activity and promote gene expression is when you add an acetyl group to these amino acid residue, what it does is that it reduces the overall positive charge of the histone tails, right? And you remember the DNA is negatively charged. So by reducing that affinity to that negatively charged DNA, you're kind of repelling or weakening the DNA nucleosome association, okay? In addition, these acetylated lysines serve as binding sites to recruit different proteins, right? Activators or repressors to that site, okay? And that's how they're able to regulate gene expression. Similarly, methylation, right? So methylation is not necessarily, adding a methyl group to the DNA doesn't necessarily involve changing the charge of the histone tails, but it just kind of increases the bulk of the histone tails. So it becomes more bulky, and because it becomes more bulky, it also helps to make that DNA less accessible, okay? And histone methylation, as I mentioned, is linked to both activation and repression, depending on which lysine gets methylated and how many methyl groups are added to that site. The main enzymes that's involved in histone acetylation is histone acetyltransferase, or HATS, and they're the ones who adds this acetyl group to that amino terminal, right, to that amino tail. This process is reversible, and it needs to be reversible, right, because you need to have that dynamic regulation of the DNA, right? You want to make it accessible and inaccessible depending on the gene, depending on the condition, depending on the developmental stage. So you want to be able to reverse this modification, okay? And the enzyme that's involved in removing this acetyl group to reverse that acetylation is histone deacetylase, or HDAX, okay? Similarly for methylation, this is also a reversible process. Histone methyltransferases direct the methylation, right? So they add a methyl group to that amino group, amino terminal. And a histone demethylase, and in this case, LSD1, which is a lysine-specific demethylase, removes that methyl group from that group. Again, highly reversible process, and it needs to be reversible in order for you to change the regulation of a gene when you need to. For phosphorylation, which we're not gonna go into too much detail on, you can phosphorylate serine and threonine, and this is also a reversible process. Protein kinases, which you guys remember, are involved in phosphorylation, right? And you can have these protein kinases that will phosphorylate either serine or threonine, but you can also phosphorylate tyrosine and other amino acids, but these are the ones that have been best studied. And this phosphate group can be removed by protein phosphatases. Okay, so again, another highly regulated and reversible process. As you'll see, that these three different types of modification can happen in an order and a particular sequence to regulate gene expression. We also have a range of other post-translational modifications that can be made to these histones that will regulate gene expression. There's not as much known about them, but they do exist, and just so you know that there's other modifications that can be made, and which is ubiquitination, glycosylation, carbonylation, or simulation, right? So you're adding these groups, like a small sumo group, for example, or a small ubiquitin-like protein to these particular lysines as well. And they also play a role in gene expression. But the ones, yes.
So going back a bit, just question I had. So the acetylation, I know that when we were going directly to the lysine and everything, the methylation, what is it specifically about adding on the extra bulk that helps to determine whether it's activated or repressed? Because if the extra bulk's added, wouldn't it just make the tail longer? Or
so when it becomes bulky, the chromatin becomes a little bit more compacted, right? So it's not as loose, it's a little bit more compacted. And when you compact the chromatin, right, you're making the DNA less accessible. And in addition, because it's compacted, any of these modifications are going to recruit proteins to that site. When you recruit those proteins to that site, that helps to even increase that compaction even further, right? So you're really making the DNA less accessible in that context. In the case where you have a methylation that is involved in gene expression, right? So the tail gets modified, it will recruit really an activated protein or a chromatin remodeling complex that can really remodel that chromatin and make that region more accessible. Yeah, but for the terms, in terms of repression, it's really because it's more bulky, it's just more compacted.
Okay, thank you.
Mhm. Yeah. So this is a nice summary to think about the different ways in which these different modifications can regulate gene expression. And you can see that, for example, you have your histone H3, you have, all these different modifications. Depending on the modified state, you can have different meanings, right? Gene silencing, heterochromatic formation, right? Meaning that you really have a compacted region. Gene silencing, lots of transposons are often associated with these regions, right? Where you really want to keep the gene turned off. Depending on, here, you're also having this lysine 9, which is it's trimethylated here, gene silencing. It's acylated here. You have gene expression, right? And this is really one way you want to think about it. Another lysine that gets trimethylated, also involved in gene silencing. So it really depends on the residue that gets modified on different histones. 1 second, Sheila, I see your hands, have your hands up. Let me just finish this last part. So when you think of the histone code, you don't want to think of it as a mutation or anything that's affecting the DNA base sequence, right? The histone code is an epigenetic code, okay? It's not affecting the base sequence, but it still can alter gene expression, okay? So a modification is being made, but it's not changing the base sequence of the DNA, okay? Yes, Sheila.
Okay, so is it either or methylation and acetylation, or can both be occurring? Because I can see the diagram like on part A, and it has like MNA, so they can both occur simultaneously?
Yes, so
Okay.
you can have multiple modifications from multiple different residue on these histone tail occurring all at the same time. With that said, there's an order, right? So one happens first, the other one happens after. So there is you're not going to go into a whole lot of detail about that, but you have to have an order, right? So in some cases, lysine 9 needs to be acetylated first before this trimethylation can happen. But they can happen they can happen, I guess, think of it as the same time or in the regulation of the same gene, right? And you'll see that actually a little bit later on in some of the slides where you're having this stepwise modifications that are happening in order to regulate a single gene promoter. Yeah. Does that answer your question?
Yes, I think so. But for like a singular lysine, like K9, how it has M and A.
Mm-hmm. Yeah.
That's what you're indicating, right? Like,
so both of them. So this lysine 9 can have both acetylation and methylation happening at the same time. Acetylation will happen first, and then that lysine will get methylated after, right? So that can happen at the same time. You can also have this aspect, right, where you have a lysine here, and then, few lysines upstream, you have methylation happening. So both of these contexts can happen, or you can have this happening at the same particular lysine.
Okay, thank you.
Mm-hmm. So someone asked, how do we know that the middle line in B is involved in gene expression? So this is just based on tons and tons of genetic studies to show the function that when you have lysine 9 acetylated, you often have gene expression. When you have lysine 4 trimethylation, you also have gene expression. So this is just based on functional studies that has been done. And you'll see that actually a little bit later. Okay. So you will need to know, do you need to know this? You will need to know this depending on the question, right? And the context of the question. As I mentioned, you need to know one acetylation, what of the acetylation marks and one of the active marks and one of the repress marks. activation is pretty easy, right? Because you know that acetylation is often associated with activation. So one of those marks, the three that we just discussed. And these are just a subset of marks. If you are maybe in the discussion, maybe Israel will tell you guys about many other different types of acetylation or methylation marks. Whatever marks have been discussed or we have discovered, they are associated with either gene activation or gene expression. Some marks we don't know enough about, so you might often see it being referred to as being either an activator or a repressor or not. Okay, so it just depends. I mean, there's tons and tons. The table I showed you is not even half the number of different histone modifications that occur within the cell. Okay, this is just a subset. Yes, Christian.
Yeah, I was just curious. Just looking at the table, I see that it's like histone for like acetylation. It's histone 3, lysine 9, AC.
Mm-hmm.
Should we make sure we know the AC2, or what is that specifically? Is that just telling us specifically it's acetylation?
Yep, it tells you that's acetylation, and that's absolutely important because you can have, I mean, if you say just histone is 3K27, I don't know whether you're referring to methylation. I don't know if you're referring to acetylation or phosphorylation, right? So that's absolutely important that you remember to add that AC or the methylation, right? So lysine, if you were to look at H3K4, right? There's 3 different versions of methylation, right? All 3 of these, whether it's mono methylation, dimethylation, or trimethylation, can be associated with activation. Mono and di is associated with repression. So if you're going to tell me that you have a repression mark and you just say H3K4, I don't know what you're talking about. If you also say H3K4 methylation, I could, just like you would assume that I know which one you're talking about, I could assume that you're talking about, oh, maybe you're talking about this one, which isn't repression, right? So you want to be very specific about the marks. And you'll also see, again, later on, why you really need to know which modification is being made, because there is, as I mentioned, there really is a stepwise process to how these things actually happen, right? So the proteins recognize that that lysine at that position have 3 methyl groups versus 2 methyl groups, right? I mean, they really do recognize that, yeah.
Okay, so then those extra marks, then that really does make a difference because different marks would have different results and such. Okay.
Absolutely, absolutely, yeah. And you'll see, we'll talk about the proteins. There's a protein that's reading these marks. So they're just not occurring and then just there. There are proteins that actually look and see, okay, you have 2 methyl groups, you have 1 methyl group, so we need to recruit this protein or that protein, right? So it's really stuff that has actually known how these interactions happen. Okay, yeah, and that's what we're going to get to, right? You have all these marks. How do you know what is happening, right? And how do you interpret these marks? We talked about the fact that we add the marks, right, to these histone tails, right, to the residue of the histone tails. The group of proteins that are responsible for adding the marks, the modification to these different amino acid residues, are often referred to as writers, right? They're writing the mark, right? They're putting the mark there. This is the mark. These marks that are added get read by another group of protein called reader proteins, okay? So these reader proteins look at these different modifications and say, okay, this is an acetylation that's present. And because we have an acetylation present, we're going to recruit all these proteins and it will be involved in transcriptional activation, or it will be recruit proteins that will be involved in transcriptional repression. Depending on the process it's involved in, right? It could be involved in some kind of DNA replication or DNA damage repair. Again, these are just interpretations that these proteins make, and then provide and then recruit the right proteins to direct that gene function. Okay. Once they're read and whatever activity needs to occur, the marks will then be removed. Okay, so these are called erasers, right? So the erasers, which are the HDACs or the demethylases, will come in and remove those marks, right, from that gene to get it back to the original state. Okay, all of this is happening before, this is actually happening before transcription can happen, right? Before the transcription, before TDP, before DNA polymerase can even get access to that DNA, you need to have it accessible, right? So that chromatin needs to be accessible, be made accessible before anything can occur. This is a discussion that's up for debate, right? A chicken and egg question. Can they happen simultaneously? Perhaps. Can it happen where maybe the transcription machinery gets to a region that's not accessible and recruits other proteins to make it accessible? We don't really know. Most of what we know is that you need to have your DNA, you need to have that promoter region accessible in order for everything to be able to sit on that promoter and make that mRNA. Okay, and in order for that to happen, you want to be able to remodel the whole chromatin and be able to do that. Mm-hmm. So these are the three groups of proteins that are involved. So writer, reader, eraser. A lot of proteins will have double function, right? So some proteins might be able to read and write at the same time. So it just depends on the protein. So you can imagine that you have your DNA wrapped around your histone core protein. This is your nucleosome unit. You have the modification that's represented by these little blue dots, right? Reader protein comes in. It's a huge protein complex. It's not a single protein, it's a huge protein complex. Comes in, reads these marks and say, okay, oh, this is probably acetylation or methylation. Let's recruit another set of proteins that will help to regulate the expression of the gene that's downstream, okay? So it's really an interaction, protein-protein interactions with these histone modifications that helps to regulate gene expression. Post-translational, all of these modifications are post-translational modifications, right? You're modifying the amino acid on those histone tails, okay? When you're pre-transcription, meaning, so Shayla mentioned that it was post-transcriptional. These, you could say it's post-transcriptional, but it's post-translational, right? Everything is happening after transcription. It's happening after all of the post-transcriptionals. So you have the protein. You're modifying the amino acid in the protein. They're post-translational modifications, okay? So that is the modification that's being made. Okay, so you're modifying these histone core protein tails. Yeah, so I hope that that needs to be clear, right? Okay, okay. So yeah, so depending on the different type of reader proteins, the two main groups that we are aware of are these bromodomain containing proteins or chromodomain containing proteins. These proteins within this protein family, there's 20, 30 different members of that family. We're discovering more and more every day. Also the chromodomain containing proteins that can function as reader proteins, right? HATs, which are the histone acetyltransferases, right? These are the ones that write the protein. They can also function as readers as well, right? So they're also able to read a mark as well. And they're also, because they also have domains in that histone acetyltransferase that can be, that are considered bromodomains, right? So they're able to also read those marks as well. And it's the same for chromodomain containing proteins. Some of the histone methyltransferases or histone methylases are able to read the marks as well. So together, having those two writer and reader complexes working together, they're able to spread that established chromatin state along the chromosome, okay? So once it gets established, based on the modification, based on the mark that's being read, it will spread it along, and you have a region within that chromosome now, or that chromatin now, that then gets modified and really loosening up that region so that the DNA becomes accessible, okay? Yes, Christian.
So then it just requires the one regulatory, and then and the one writer, and then constant readers can be connected because the DNA is loose.
So when if you have a writer, right? So imagine you made a mark, right? So here in this case, right, you have a regulatory protein for some reason, recruits this writer protein, it adds the mark, right? Modifies the mark. The reader protein comes in, right? And this reader protein could be attached to the writer in the case of the hats, or it can be an additional reader protein that gets recruited by this writer protein, right? Comes in, reads the mark, right? And it says that this is what the mark is. Because these two are interacting, protein-protein interaction, this remains associated, right? The writer goes to the next nucleosome, the next histone tails that's adjacent, marks it. The reader can then read it, and it can keep going from nucleosome to nucleosome that's adjacent to each other, right? The tails are going to be sticking out, so it can do that and can really spread that particular mark. So
and does it keep going, or there's like site?
so it will keep going until it. So there's a point where it cannot go any further. What determines that really depends on how long a promoter region is, right, within a gene. So it really just depends on the regulation of that gene, how vast an area you need to make accessible, right? Some promoters regions are longer, some promoter regions are shorter. It really just depends, right, on the gene. But at some point it stops. It will not go into the gene region or the coding sequence in that case, right? You just want to make accessible the promoter region.
And you said that as the writer's going through and the readers are attaching, it's loosening up the DNA a little bit so it can be read a little better?
Yes, it's depending on the modification mark though, right? If it's acetylation, it will loosen up the DNA, right? Because it will recruit chromatin remodeling complexes that will help to loosen the DNA. If it's a methylation mark that's associated with chromatin, like making the chromatin more condensed, right, more repressed, then it will be reading along and it will be compacting that chromatin more, right? So it just depends on which mark you're referring to.
Okay, awesome, thank you.
Okay. Mm-hmm. Okay, so Israel wants to clarify again. All of these events are happening to the histone tail. The histone is a protein. The amino terminal has amino acid residues. You're modifying the amino acid residue. This is occurring after the post-translationally. Okay. I mean, you're modifying the protein, right? It's a post-translational modification. You're not modifying the DNA, right? The DNA is being modified because you're altering how that histone protein is wrapped around the DNA, right? That's what that's how that gets modified. But histone modification is modifying the protein tails. So it's a post-translational modification. Okay. So then you have this series of events where you have, readers and writers, which is part of these protein complexes, which can contain, again, it's not a single these examples are always showing single proteins, but it's not a single reader or writer. You can have multiple readers and writer within a complex that's there establishing those marks, those histone tails, right? So it could be many different additional readers and additional writers. This is really just a simplified graphic of what's really happening. Okay. So depending on what's happening with those chromatin remodeling complexes, you can decondense the chromatin, making it more accessible, or you can condense the chromatin, making it more compacted. It really just depends on the mark that's there. And as you mentioned, you also have these sequence-specific DNA binding proteins, such as transcription factors, that work together with the nucleosome remodelers, the histone modifiers, to really make that DNA more accessible. So if you're looking at this graph, for example, you have your 30 nanometer chromatin fiber, where that means that it's compacted. And you're going from this compacted state to this more loose state of DNA. And you, in this case, this is at a particular point where you have a DNA binding protein, maybe an activator or some type of protein, that recruits this histone, this chromatin remodeling complex. This chromatin remodeling complex, right, they remodel the chromatin, right, either by sliding, eviction, or any kind of movement of the DNA out of the place of the histone where it was, right? This remodeling exposes now another piece of DNA. This DNA binding protein can come in, bind to this site, right? Binding to this site, now it's able to recruit HATs, which are histone acetylases. These histone acetylases says, okay, I need to modify the tails of these histone. It adds these acetyl groups to the tail of the histones, right? And again, by adding these acetylated tails, you loosen the DNA, right? Because acetylation reduces that positive charge of the histone, the association between DNA and the histone become less strong, okay? So it's a series of combination of proteins, DNA binding proteins binding to DNA and recruiting these different chromatin remodeling, or I would say, as I said, nucleosome remodelers, right? Because you can have chromatin remodelers, or you can have histone modifying enzymes. And this is exactly what happens in the cell, right? Depending on the gene that needs to be regulated, and also the condition within the cell, right? This happens for some nuclear receptors. For example, the thyroid hormone, right, which involves interaction with ligands and receptors in order for the function to happen. So in that case, depending on whether or not you have the thyroid hormone present or not, will alter the expression of the gene. Okay, so this is a schematic that you have here, right? So the names of the different acronyms that you can see here. So you have a thyroid hormone receptor, retinoic acid receptor, often form a dimer. These dimer will bind to elements within the DNA known as thyroid hormone response element, or TREs, okay? So chromatin is unperturbed. This is the state that it's in. These proteins, will recognize this site and bind to it when it's needed, okay? So for example, if you were to have a situation where there's no thyroid hormone in the cell at all, you don't need to express this gene, okay? You really don't need to have the gene expressed. You don't have any thyroid hormone that's being present. So you don't necessarily need to express this ligand. So in that case, when you don't have thyroid hormone, you have this dimer that will bind to the site, which it normally would, right, to the TRE element. So what it does in this situation, it recruits histone deacetylases and also co-repressors, okay? So it's recruiting these enzymes to help to repress the chromatin state. Okay, so it's really compacted because you don't want any transcription because there's no thyroid hormone available. So you don't need to be able to express that. Yeah. So in the case where you have transcriptional coactivators, I mean, when you have thyroid hormone, you will recruit transcriptional coactivators, histone acetyltransferases, right? You have your TAFs. You guys remember the TAFs are these proteins that Tadabox binding proteins that also have histone acetyltransferase activity. And you can get recruited here to this site. You modify the tails. You kind of loosen the chromatin, and you can then promote gene expression. Okay. So depending on the situation, the gene that you want to be expressed, depending on the condition in the cell, maybe it's a different developmental stage, maybe it's in response to stress, you can have a particular gene being regulated either of two ways. Okay. Okay, so I'll pull the slides up because computer crash, so we'll just go with, okay, let me just find slide where we're on. Okay, so that's really the situation depending on what's happening in the cell. In the case of other genes within the genome, right? So we have an interferon gene, which is involved in immunity in the cell, right? So they are turned on depending on an infection. So if you have an infection, you will express this particular gene within the cell. In order for this gene to be transcribed, you need to have a series of modification that occurs after a viral infection has occurred. Okay, so depending on what virus you get infected with, and if it's related to this particular human immune receptor, then you will have you will need to turn this gene on, right, to produce it so that you can deal with that infection. Very basic schematic, right? So it's showing you a nucleosome, histone tail sticking up. This is the Tadabox, right? You have the enhancer element. We talked about the enhancer elements, which helps promote gene activation, right? So activators are bound to these enhancers. Similarly, you have an infection. This enhanceosome assembles at the promoter. An enhanceosome is just a series of activator proteins or proteins that are involved in enhancing gene expression. These proteins are able to recruit histone acetyltransferases, right? So they recruit this huge GCN5 complex. So to acetylate histone 4, lysine 8, and histone 3K9. These are the two lysines that they first need to acetylate in order for this entire gene to be turned on. Okay, so these two needs to happen first. This activator will also recruit a kinase to phosphorylate serine 10 on H3. Okay, and in the event of doing this, as you can see here, you also will acetylate lysine 14 at the same time. So this activator is doing all of these modifications. Okay, it's recruiting a kinase to phosphorylate serine 10, and it's also acetylating lysine 14. Okay, so that's the next step. Okay, the acetylated H3K4 that happened initially will then attract this SWI/SNF complex, right, which is your chromatin remodeling complex that gets recruited. This chromatin remodeling complex will then remodel the chromatin, right, to the point where you see now you give enough space here for this Tadabox that was there initially. You give enough space that TF2D can bind to that Tadabox, right? And this is really how this gene gets expressed, okay, by the series of these different steps that's happening in sequential order as well. Is that clear? So I broke these down. This is a little bit, there was more information. I broke these down into these 5 steps, right? Because I think it's really easy to understand just by looking at the different 5 steps that's happening in these orders. Okay. And the way that they figure this out is by, I don't have my time, okay. The way that they figure this out is by doing another protein DNA interaction assay. This protein DNA interaction assay involves protein interacting with the chromatin. So it's called chromatin immunoprecipitation, okay? And the idea behind this is that you have your proteins, yeah? Protein is gonna be bound somewhere to your DNA, and you can then isolate this protein. And by isolating this protein, you're also isolating the DNA that's associated with the protein, okay? Once you isolate this DNA that's bound, the protein that's bound with DNA, you can then remove the protein and just look at the DNA. And depending on the technique you're using at this step, you can either amplify that DNA by PCR to see what it is and run it on a gel, or you can sequence it to look at the sequence, right? So you can do chromatin immunoprecipitation, or CHIP, we often refer to this as, followed by a PCR, and it's called CHIP PCR. Alternatively, you can do the chromatin immunoprecipitation followed by sequencing, and that's called CHIP sequencing. The differences between these two really depends on what the question you have. If you're just interested in a single gene, then you just do a PCR for that single gene, right? And that's exactly what they did. They were only interested in human interferon B promoter, so they just did a PCR for that promoter region to see what proteins are bound to that promoter after infection with this virus. If you're interested in all of the genes that could possibly be regulated by this particular protein, then you will do a sequencing to see what's happening genome-wide. And that's exactly what they did in these experiments to look at that interferon B promoter. They did a CHIP followed by PCR technique. You're looking at a gel. You're looking at the protein that's associated with that particular DNA at a given time, okay? The top is showing you time in hours, right? From 0 post-infection time points, right? So 0 to 24 hours after infection. And you also have the different possible modifications that's happening at that DNA, okay? So the protein that you're looking at, so you're looking to see, is there an acetylated H4 lysine 9 happening at the promoter? If you see a band, that means that it's there, okay? Because you isolated the DNA that's bound to this histone modification mark, and you did a PCR, so you're seeing a band. Okay, and you can see when it's happening, right? This was also pretty powerful for them to see when the events are occurring, right? So you see very early on, you have H3K4, 3 to 8 hours that's happening. K12, for example, is only happening much later at 6 hours, right? Phosphorylation of serine 10 starts about 3 hours, but really peak around 6 hours. When that's happening. And then you remember that you will have H3K9 happening, very early on at the same time with H4K8. And then this is the step that needs to happen, right? Histone 3, lysine 14 acetylation, which occurs 6 hours after the infection. Remember, this is what actually happens that helps to recruit TF2D. TF2D, what's binding to that promoter is a TBP protein within that TF2D, right? So you can see only when this has happened can you actually detect this TF2D protein, not before, okay? And when this happens, you're looking at gene expression. Now you have mRNA, right? Because your TF2D is bound via this TBP protein, and now you can have expression of the gene. Again, this is an experiment that was actually done, that was actually taken from a paper where they actually did this, right? So they can really see when these different modifications are occurring and when is the final point before you're able to actually transcribe the gene, okay? So these are all the different modifications. Input is really just to make sure that you actually have DNA present within the cell, right? So that's what this input is. It's really just a control. Okay. So understanding the steps either by understanding what's happening here or the step that's happening here, you would be required to know, right? I mean, because it really tells sequentially what needs to happen within a cell before a gene can be expressed depending on the context. Okay. So in terms of thinking about everything that we've talked about, understanding what is chromatin, right? That you need to have this chromatin modified in order to regulate gene expression. This is how eukaryotic DNA is comprised of chromatin, right? It's DNA filled with proteins. What is a nucleosome unit? What are the histones, the core histones? What happens at the histone tails, right? What are some of the chromatin remodeling complexes? Really understanding what is happening here, really directing which mark might be associated, one of the marks, I shouldn't say which one mark associated with activation, one mark associated with repression. Understanding the steps of how this interferon B promoter gets regulated, again, it's 5 steps. If you understand these 5 steps, that's really all you need to know. You don't necessarily need to focus on this technique. I mean, this technique is really great at highlighting the different steps. But if it's something where you're, maybe you don't get it, you don't have enough time to discuss it, this is really representing exactly what is depicted here graphically. Okay, it's just the experiment that that made it possible for them to actually draw this model of what's happening at that interferon B promoter. Okay, I'll take a break. Give you a poll so that you can, if you have any questions, you can think about it. Before we get on to DNA methylation, which is another way that you can regulate gene expression. And since I'll just try to make this short so that you can check that. Let's talk about chromatin remodeling complexes. Are you able to see that poll?
Yes.
Okay. So all chromatin remodeling complexes have what activity that is necessary for their function? Okay, let me end it here. 78%. Do you see the results?
Yes.
Okay, anyone want to take a go at this? So I'll start from the bottom going up since that was the least popular answer. So all chromatin remodeling complex have this particular activity that is absolutely necessary for your function. Transcription factor binding, not necessarily, right? They don't necessarily bind with transcription factors. I mean, they could, but it doesn't necessarily do that. DNA binding, chromatin remodeling complex don't necessarily bind to DNA, right? They interact with other proteins that might be bound to DNA. Repressor binding, it depends, right? It could be that they could bind to a repressor or get recruited by some kind of repressor molecule. Maybe it's, depending on the chromatin remodeling complex or the protein within the chromatin remodeling complex. ATPase activity, this is really the definition of a chromatin remodeling complex, right? It is ATPase dependent. You need to have ATP in order for this to work. Yeah, these proteins cannot function without ATP. So that is something that you want to remember. Histone binding, a lot of you said histone binding, and I can see why you would think that. But remember, these chromatin remodeling complexes are being recruited to these histone tails by a protein that's bound to the histone, right? Is that clear? So your reader protein goes in, right? Your reader protein is going to go in, and your reader protein is what's bound to that, what's recognizing that histone modified mark. And that reader protein, which is bound to that histone, is what is going to be recruiting that chromatin remodeling complex. So you can think of it as histone binding as an indirect histone binding, but it's not actually binding to the histone. It's there at the histone because it's associated with that, whatever protein is there at that histone. Right? So yeah, all this is just a function of these proteins, right? Based on, again, genetic studies and everything that we've done. You need to have ATP, you need that energy for these chromatin remodeling complexes to work. They're dependent on ATP, they cannot function without it. Yeah? So they're often referred to as ATP dependent chromatin remodeling complexes. Correct. It's B, yeah, it's ATPase, yes.
Can you go back to the mechanism of replacing histones? That was like one of the first slides.
Which one was it?
It was like
This one? Or?
No, not this one. Maybe
This one?
no, not this one. Replacing the histones with like another, like different version. Yeah, this one.
Mm-hmm.
You see, like it doesn't say it's ATP dependent.
But it says here. Yeah.
Mm-hmm.
Yeah.
That's for all of them, all different.
All of them, all chromatin remodeling complex. And I think I also said that all chromatin remodeling complex require ATP, yes.
Because like for the first two mechanisms, the diagram, it says like, we use ATP, but for the last one, it doesn't say. So
yeah, it's just, this is just a graphic, yeah, but it's
okay.
they're all ATP dependent, yes.
Mm-hmm.
Mm-hmm. Yeah. Okay. Okay. So the last few minutes, we'll get started on cytosine methylation, right? So we talked about chromatin structure. There's also other types of regulation that can happen at a gene promoter or even along the gene body that also affects the expression of that gene. One of the mechanisms that has been well studied is called cytosine methylation. And this is where you're adding a methyl group to the 5th cytosine. In the 5th carbon of cytosine to regulate gene expression, okay? This methyl group gets added by an enzyme called DNA methyltransferase. And depending on where the methylation occurs, you can direct gene expression in a different way. And we'll discuss that in a minute. So most often, initially when cytosine methylation or DNA methylation was discovered, it was discovered because it was involved in gene repression or gene silencing. Okay, so this is what we taught for a very long time, that DNA methylation is associated with gene repression. So DNA methylation of the promoter regions usually will inhibit transcription. The regions that are more highly methylated, meaning that it has lots of methyl groups added, will have a more compact chromatin, and that's one of the reasons it's associated with gene silencing. You can have 2 different versions of DNA methylation. There's what we refer to as trans-symmetrical and then non-symmetrical patterns. So in plants and animals, you have these what we call GC sites. Depending on where the information is coming from, you can see it being written as CG site or this CPG, where they show that there's this phosphodiester bond between the 2 nucleotides. Okay, but it's a GC site, meaning that, and this is symmetrical because you have a GC at the bottom. Okay, so there's symmetry here. In a plant, we also have a different type of DNA methylation that we observe as well, and these are called in a symmetrical format, where you have a C, and instead of a G right next to it, you can have any nucleotide. Okay, these are also symmetrical because the bottom strand is going to be kind of the same, right? There's also going to be a C, whatever complementary nucleotide, and a G. But you can also have non-symmetrical DNA methylation, where you just have a C and any other 2 nucleotides that you can have. Okay, and that's kind of non-symmetrical. So you can have either of these 2 versions. And yeah, so the question, plants are the only ones with non-symmetrical DNA methylation. It's definitely not in humans. I don't recall if we can find that in yeast or maybe fungi or neurospora, but for the most part, this is often found in plants, and you'll see the reason why that is, okay? So depending on where that methylation happens, we now know that you can regulate gene expression in either of 2 ways. So initially, when it was discovered, you had, and this is what these little circles and the different types of methylation. If it occurs in the promoter region, you will have gene silencing. However, you can also have methylation because it's a DNA sequence. You're adding a methyl group to a cytosine. If that occurs in the gene body, right, or a coding sequence, it actually is able to activate gene expression. We don't know why, but we have observed this, right? There's many different hypotheses why this might be happening, which we don't need to get into, but this is an association that we've seen. Okay, so promoter region, gene silencing, very classic what's been studied, but there's also cases where you can have gene body methylation within the coding sequence that's involved in active gene expression. So I'll probably stop there. I just wanted to get to Christian question very quickly, and I don't. So yeah, so the reason that we know that only plants we've observed so far have this non-symmetrical or the CHH type of DNA methylation is because of the enzyme that makes that modification. Okay, so so far we've only found these enzymes, these chromomethylases that's involved in maintaining this non-symmetrical methylation in plants. Maybe in the future, as we're studying more and more genomes and able to understand more what's happening in other genomes, we might find it in other organisms, but for now, this seems to be primarily found in plants. Yeah, I really hate having to say something is only found here and there because we just don't know. But so far, from what we understand, you only find this CHH present in plants, and that really has a lot to do with the enzyme that's involved in maintaining that particular type of methylation. Okay, so I'll stop there since we're already over time, and I'll stick around if you guys have any questions about anything.
I had one quick question, Professor.
Yes.
For the cytosine methylation, you were saying that in plants and animals, the gene bodies, it can still translate through. So if the methylation's only in the promoter, is when it inhibits the transcription?
Yes. Mm-hmm.
So if the methylation's anywhere else along the gene body, it doesn't matter. But if it's specifically in the promoter, that's when it interferes with the gene being read.
Okay, yes, but let me clarify. So if it's GC methylation, okay, so GC methylation in the promoter, you have silencing. GC methylation in the gene body, you have activation. You could have gene body methylation of one of these two flavors, and they're actually involved in gene repression, but it's a different mechanism that we're not going to get into. Yeah. So
okay, so
here,
this is just specifically CG.
Specifically G, yeah, exactly, the CG methylation. Okay.
Okay.
Yeah. So thanks for actually bringing that up so that we can clarify that. Yes.
Okay. Thank you.
So there was, I had another one of these little gene graphs here where it had if you're interested in this kind of stuff, you can look at this paper where they had another graphic showing within the gene body CHG and CHH, and that's involved in repression through a different mechanism, a post-transcriptional mechanism that I didn't want us to get into. So yeah. Your hands are still up, Christian. You have another question, or?
Oh no, sorry. I was writing stuff down.